Our story starts at the beginning of life, where a mesmerizing process takes place, the embryogenesis. Here, a single cell, the zygote, will be potentially able to get an entire organism. After the phase of cleavage, where cells divide rapidly until they reach the so-called morular shape, a process breaks the symmetry and polarizes the outer cells. They consequently dismount themselves along the boundary, forming the trophectoderm. These cells express a particular gene, CDX2. In the meanwhile, a cavity is formed, the blastocoel. At this point, it is possible to observe a certain peptide pattern of Nanon and Geta-6 gene expression, which then characterize the two next-generated tissues, the epiblast and the primitive endoderm, formed after a phase of cell's movement. Our purpose is building a model which is able to reproduce this phenomenon, performing numerical simulations which take into account the interplay between mechanics and the gene regulatory network. Let's suppose our cell is a sphere, which acts as if there were a spring inside. If it is compressed, it will exert a force opposite the compression. Let's add adhesion properties which stick the cells together and make their relative movement difficult. On the other hand, within the cell, chemical reactions can occur, and a gene expression prevails. Different gene expression yields different mechanical properties of the cells, such as adhesion. In this fashion, running the simulation, we notice that the reflector information is well explained if we add an external signal whose intensity depends on position. This process is responsible for the activation of CDX2 on external cells, and the particular adhesion properties drive the cells in the correct position. However, if the properties of our model are applied to Nanon and Geta-6 gene expression, they are not sufficient to explain the formation of epiblast and primitive endoderm. Cells do not reach the correct position. Even if our model cannot explain the whole complexity of embryogenesis, the results of our simulations could be the starting point to project new experiments to solve this puzzle.